All right, this week we're going to cover prokaryotic cells and viruses. So this actually has real life implication as to what's going on right now. I mean, as far as the virus route goes. So we're going to do some basic terminology and parts of a bacteria cell, then parts of a virus. All right, we already discussed this a little bit earlier as far as prokaryotic cells go. They're very simple. They don't have what are called membrane-bound organelles. They have a nucleoid region, and then yes, they can have layers as far as a cell wall and a capsule and a cell membrane. They do have ribosomes. It says they're a food granule. We did talk a little bit about this. Bacteria can exchange what are called plasmids. These little circular pieces of DNA that exist in real life. And anyway, that's a lot of times these plasmids will carry what are called beneficial or luxury genes. And bacteria do have flagella. And this guy here actually has something called fimbriae. So we'll go through all that. And this is not a very long section, but this is something to start off with. When you study bacteria, the first thing you do, if you're a microbiologist and you find something somewhere growing and you want to do some experiments with it, or you want to try to classify it, figure out what you're looking at, what you do is you grow it on a petri dish. You then get a small part of it, you put it on a slide, a glass slide, and then you stain the cells. And you look to see what is the cell shape and the arrangement. There are three basic shapes for bacteria. Cocci, which are circle-shaped, bacilli, or bacillus, this is a singular term, rod-shaped, or spirilla, or spirillum, which are S-shaped. And here's some examples. That says, now that's, there's cocci. Could probably get some better pictures than this. Cocci, this is actually a group of, looks like 2 or 4, they're in packets. Here are the rod-shaped bacteria. Vibrio, or comma-shaped. Here's the spirillum. You see they're kind of like little squiggly shaped. These are spirochetes, which is what syphilis, the shape of syphilis is. And these are branching filaments that you find, a lot of time in yeast, things like that, which that's interesting that's in there. But there's 3 shapes, and then here are the arrangements. Okay, the cell shape is this, and then the arrangement depends on the species. There are some bacteria that when they divide, and they divide very simple, there's no mitosis. It's called binary fission. You have one cell and it divides into 2 cells. Very simple process. Depending on the species, some stay together in pairs. And of course, these are microscopic. You can't see bacteria anywhere. The only way you can really see it if you grow it into a colony on a petri dish or something. And then you can look at it that way. But even then, the cells are microscopic. A diplococci is a pair of 2 circle-shaped cells. Gonorrhea is a gram-negative diplococcus. Streptococci is a chain of cells. Staphylococci is like grape-like clusters of cells. A tetrad is 4. And this is Sarcinae, which is a group of 8. And they're circle-shaped, of course. Let me see if I didn't. There's some... Okay, there's some itty-bitty bacteria micro. I thought I had better pictures than this, but this looks like this would be, they almost look like they're in 2s. They could be in 4s. You see they're circle-shaped cells. This is called a streptobacillus. It's a rod, a chain of rods. But if you had a chain of circle-shaped cells, it'd be called a streptococci. Bacillus, diplobacilli would be 2 rod-shaped cells. Strepto is a chain of rod-shaped cells. palisading is where they're kind of all unevenly stacked on top of each other, and then sporilla, a little misleading there, but sporilla is not a bacillus. It should be over there as far as its own category, and they're not found in groups. And then Vibrio, like I was saying, those are comma shaped. So these guys, all right, and these other ones like the spirochete, sporilla, they're not really found in groups. Now this is very sciencey. Bacteria have cell walls. Human eukaryotic cells do not have cell walls. Ours is a cell membrane or plasma membrane. And there's actually a method of staining, and that's what you'd have to do with these. You would take a sample off a petri dish, you would stain the cells, and then look at them under a microscope. And you have to have a microscope that goes up to about 1,000 magnification to even see them. That's how small they are. And that's what you would do to determine these. Now, the cell wall. There is a guy who came up with a way of staining to determine. You could almost put bacteria into two categories. Ones that are gram-negative, and then ones that are gram-positive. And here's the reason why. I like simple diagrams. This one's very simple. This is a gram-positive bacteria, and you probably recognize this. So remember the bilayer or lipid bilayer on the bottom? There's the hydrophilic heads and the hydrophobic tails. That's a cell or plasma membrane. Bacteria have that. Human cells have that. That's where we stop. But bacteria have a cell wall. Okay, and these are categories. Not every single bacteria falls under these categories, but a big percentage do. So gram-negative bacteria, their cell wall has two layers to it. There's a layer of this stuff called peptidoglycan, and that's important here in a second. And then they have this outer layer that's called a lipopolysaccharide that's almost functions like a cell membrane. So things that cross into gram-negative bacteria would be different than something that could get inside of a gram-positive bacteria. Gram-positive bacteria have a big, thick layer of what is called peptidoglycan. Peptidoglycan is a large molecule, and interesting enough, it's the target of penicillin. So the guy that discovered penicillin back in 1929, penicillin is produced by Penicillium, a fungus, and what it does is called an antibiotic. It kills. Bio means life, so an antibiotic is going to kill life. But antibiotics are chemicals produced by fungus that kill other microorganisms. And penicillin prevents peptidoglycan from forming. So when you have a bacteria infection, when the new cells start to divide and they're going to start building their cell walls, penicillin prevents that. So penicillin is really good at killing gram-positive bacteria. It's not as effective on gram-negative bacteria. And that's a whole other story for another day. But gram-negative bacteria are not killed as easily by penicillin as gram-positives are. And if you're wondering, okay, what are some familiar ones? Staphylococcus, Streptococcus, any of those are gram-positives. You've probably heard of Escherichia coli or E. coli, Salmonella. All those are gram-negatives. So here's a little bit about them. So there's two basic cell walls, gram-positive, gram-negative. And like it says here, gram-positive bacteria cells have a thick layer of peptidoglycan. They have other things too, but that's the majority of their of the cell wall is made of that. And then gram-negatives, like I was saying, they have what's called a lipopolysaccharide, and then they have this thin layer of peptidoglycan. Now, I hope, let's see, no, it doesn't look like it. So here's a gram-positive, and then here is a gram-negative. So just a little more, this is a very detailed diagram, of course. but it puts bacteria to two categories. It's really easy to do. If you find something new, the first thing you do is you stain to see what are the cell shape arrangements, and then you figure out is it gram-negative or gram-positive. Not everything can be gram-stained. Like a spirochete, that syphilis doesn't respond to a gram stain. All right, so that's just one part of a bacteria cell. Now, I don't have it here, and I don't want to add a lot of extra things that we're not covering, but there was a guy named Hans Christian Gram, and he came up with the gram staining technique. It involves crystal violet, which is purple. Acetone alcohol is called a decolorizer. And iodine is used, and then another dye called safranin that's red. And what you would do is you would put some cells on a slide. You would then flood the slide with crystal violet for 1 minute. You let it sit there. You wash it off. You add iodine next, which is called a mordant. It actually binds to the crystal violet. And basically it stains all the cells purple. And then here's the key step. You do iodine for a minute. Then you drop acetone alcohol for just a few drops. It's called a decolorizer. And that's the key step. Acetone destroys the cell wall of a gram-negative bacteria. It doesn't affect cell walls of gram-positives because they have this big, thick layer. If anything, it seals it off even more. So at this point in the staining technique, inside the cells, they're all purple. When you add the acetone alcohol, it destroys this lipopolysaccharide because lipids dissolve in alcohol. And it basically washes out the crystal violet. And then the last step of the procedure, the gram staining, you add safranin for 30 seconds. Gram negatives will stain red. That's what safranin is. They will take up the counter stain. But gram positives keep the primary stain because their walls are not destroyed by acetone alcohol. So gram positives stain purple for crystal violet and gram negatives stain red. So it's a 4-step thing: 1 minute, 1 minute, a few drops, and then 30 seconds. And you got to make the slide. You got to put some water on it. You got to put the bacteria there. You let it air dry, and then you do the staining technique. All right, capsules. Not all bacteria have a capsule. And okay, and I didn't mention this up here. The cell walls give the cell shape, allow cells to attach to surfaces. We'll get to that. But a capsule is a sticky outer layer. So you can see it there. That's what that third layer. So there's the plasma membrane, there's the cell wall, and then the super thick layer is called a capsule. Capsules are sticky. If you're working with microbes that produce a capsule and you're using one of those loops or some kind of scientific instrument in a lab, capsule forming bacteria will stick to the loop, and it allows them to stick to a surface. It also prevents them from being broken down by white blood cells. So anything with a capsule is considered a pathogen. They're hard to kill with a capsule. And plasmids carry, some plasmids, these little rings in here, they will carry luxury genes. In other words, a DNA code to produce a capsule. And bacteria can exchange those. Appendages, bacteria have fimbriae. These little guys here that are like Velcro, allows them to stick to a surface. Here's the single chromosome inside. That says plasma. And then there are other things called pili that are a little bit longer, but they're bristle-like. Flagella, yes, move the entire cell. Okay, and I don't see a mention about pili. Maybe later, but so anyway, that's a little bit about bacteria as far as fimbriae allow them to stick to a surface. Flagella move the entire cell. Here are the different arrangements. You have monotrichous, lophotrichous, which is 3 or more on one end. Amphitrachus, which are bacteria on each, or bacteria, flagella on each end, and then peritrichus, which means they're around the entire cell. Peri means around, little prefix. Right, and those are the arrangements for the flagella. Okay, we're moving into a part where you talk about how do you kill bacteria. And here is, safety levels. Once again, this is really popular at the moment with the coronavirus going around and everything. People wearing masks. But what this is, a BSL-1, these are talking about levels of safety within a building. And if you're going to work with microbes, there are some microbes that you can only work in the higher level labs. The ones that have the HEPA filters and the hazmat suits, and you get sprayed when you leave and things like that. But a BSL-1 is probably like a little college lab. Not known to cause, you have inside there microbes. You can work with them that are not known to cause disease. So minimal precautions, regular cleaners. That's like the lab at Howard College or Angelo State University, that kind of thing. A BSL-2, they are designed for handling hazardous agents, limited entrance to the facility. When work is being conducted, instruments are disposed of with care. Aerosols are handled carefully. Could maybe do that in a college as well, but these are, you're working with things like MRSA, methicillin-resistant Staph aureus. You have to be much more careful. Of course, you couldn't expose students to that. You can't expose them to something they could potentially get. But those are a little higher security. Now, the BSL-3 and 4, those are the strict ones that you see. In fact, that person there is working in either a BSL-3 or 4 lab. So all work done in safety cabinets, high-efficiency particulate HEPA filters, double doors, hepatitis, tuberculosis, anthrax, the higher end bad stuff. A BSL-4, if you work there, I guess you would go home and maybe get used to it, paranoid, but I guess they would call the coronavirus in there, but smallpox is fatal. Kills just about any person. There's no more smallpox, but it was the term is virulent. It's very deadly. So is Ebola. But you see all the different precautions. Multiple showers, vacuum room, ultraviolet light room. UV light kills bacteria, so if you work with the lights, it's just another precaution. But those would be very strict. You wouldn't want to walk in there and have a tear in your suit or something. All right, these are terms related to growth, control of growth. Sterilization means killing every single thing. An area can be sterile and be dirty. Sterile just means nothing's alive. These terms, disinfection and antisepsis. Disinfection is something used on inanimate objects like tables and things that are not living. An antiseptic is used on living skin. If you spray alcohol on a table, it's being used as a disinfectant. If you spray alcohol on skin, it's being used as an antiseptic. Physical methods of control. Now, a big thing about microbes and germs is control of growth. How do you kill them? And these are called physical methods, and then there are ones that are called chemical methods. Moist heat used to disinfect, sanitize, sterilize, pasteurize. If you don't know what that means, it's like an autoclave, and I'm doing this just like I would in class. I should have already had that pulled up. If you don't know what an autoclave is, there's one. Hopefully this will, big enough. That's not a very big one. Those are actually flasks, and that's probably some kind of media that they're making to grow microbes. But see, you close the door. It uses steam and very high temperature. And nothing can survive an autoclave. It's a sterilizer. There's the settings. 121 degrees Celsius is very hot. 20 degrees Celsius is room temperature, so that's 70 degrees. 37 degrees Celsius is body temperature, so 98.6. So that's how high that is. Pressure and then steam. Yeah, it heats up the water. The pressure forces the heat inside of cells, and some bacteria are called spore formers. You can see that. A spore is a resistant structure. Some bacteria have the ability to put their DNA inside of a spore. Spores have been known to sit for 1,000 years, and that they get inhaled, or if they're picked up and grown, they'll come out of the spores and start growing again. And they're very hard to kill, but autoclaving does. Boiling actually does not. That's an example. Boiling will still kill most things, not everything. Pasteurization is heating like wine, beer, milk to the point where you're killing the pathogens in the fluid, not necessarily everything. But anything that's been pasteurized will last longer, doesn't spoil as fast. Dry heat, incineration, very few things can handle that, but that's definitely a sterilizing thing. Nothing can survive flaming fire. Freezing actually does kill some things, but mostly it makes the cell inactive. That's why when you freeze meat, it will last so much longer, because the microbes cause food spoilage, and if you freeze something, of course it's going to slow down the cells dividing, and so food doesn't spoil. Desiccation is something that's dry or drying out. Freeze-dried food that you have to, I believe the MREs have that. I actually had one of those at one point. NASA food, that stuff doesn't spoil because it's so dry. It's the whole osmosis thing. Remember, water is drawn to areas that have high osmotic pressure, and anything that has very little water has a very high osmotic pressure, so nothing can survive that. Okay, the term communicable diseases, that is person-to-person contact. Oops, and we went to a virus there. There's a HEPA filter that's sucking up the fumes. There's an autoclave. That's an example of how Fimbre work. And then I wanted to show this, which, like I said, right now this is, it's kind of popular. An endemic, if you're talking about communicable diseases, they're passed from person to person. An endemic, you see that term, EN, endemic, is isolated cases, small areas. Sporadic has no pattern, like Ebola does that. Ebola will just show up sometimes and you'll have a small outbreak. An epidemic, now, the way it's explained to me is that involves like a country, an epidemic. It's showing there just the top of what looks like that's South America, which is a continent, but gosh, I'm geography retarded. This upper, that's a whole country in South America. And then pandemics are worldwide, which I have one of those going on right now. Let's see here. This says, example of things that, I guess these are microbes that cause illness. Rickettsia, you can see bacteria, and then you can see it says drug resistant, non-drug resistant. And there's a lot of things that over time, because of being exposed to the same cleaners, the same drugs, you have things that become resistant at some point just due to that exposure. Okay, just a little bit more. As far as disease transmission, so contact transmission, direct contact, that is person-to-person contact. Body fluids, touching, that kind of thing. Indirect contact would be like through needles or inanimate objects. There are some things like tuberculosis that can live pretty long on a countertop or something, and if you were to touch that after somebody else did, that's a form of indirect contact. Droplet transmission is sneezing. Once again, this is so relevant now. People are being told to stay 6 feet apart. As far as the social distancing part of that. These are ways you can get sick. So waterborne, foodborne, airborne. This is drinking contaminated water. This is, of course, eating bad food that's not been refrigerated. The term for that is temperature abuse. Temperature abuse, leaving food out for extended periods of time. Food already has microbes. If it's not refrigerated, most bacteria divide every 30 minutes, even sooner than that. So if you have 1 million bacteria cells, think about how many you'd have in 4 hours. You're going to have, if they divide every 20 minutes, so you're going to have a lot. Airborne are droplets that can either travel on dust particles. Try not to date this video, but the coronavirus is one of those. It's an airborne. If you're around somebody, I think they've figured out that if you're far enough away, you're not at risk. But a person who has active tuberculosis, they are, if they're infectious, if you're in the room with that person, then you've been exposed. So, all right. I'm going to stop the video after this part. So vectors are arthropods, insects. Insects that bite people and they can spread disease. Mechanical transmission is an insect running across something. Cockroaches are bad for that. Their feet are like Velcro, and so whatever they've been in contact with, is they can spread it that way. Or the biological transmission is being bitten. Well, this actually has some, so here's coughing, sneezing, here's a vector. Skin cells with open lesions, removal of blood, that's indirect contact, and this is showing there's ways. Any opening to the body is an entryway for microbes, and it's also a way for a person to spread disease as well. Wow, like I said, this is all so relevant right now, but on contact transmission, this is showing kissing. Here is indirect contact fomites. Here's food, water, biological. Sorry, the writing is so small. Hantavirus is carried by mice. That's showing droplet transmission. But all these are direct. Now that one says insect bites. So these are direct. This one here is definitely a direct contact example. Whew, there's a sneeze. You don't see that very often. Somebody actually photographed a sneeze. Yeah, these are, wow. So here is asymptomatic carriers, incubating carriers, convalescent carriers, chronic carriers, passive carriers, and that's kind of trying to show typhoid fever. Typhoid fever is Salmonella typhimurium. It ends up in the gallbladder of people, and people can be carriers for it. It doesn't make them sick after they get it, and they can transmit that to other people. All right, there are vectors. So this is biological, ferocious chicken. But yeah, bats, rabies, there's a cockroach, there's flies, stuff on their feet. But mechanical means just being touched. Biological is actually getting bitten, and they carry the pathogen in their salivary glands. So when they bite an infected person or animal, they ingest the pathogen, but it doesn't affect the vector, and then the vector goes and bites somebody who doesn't have it. And that's how it can be spread. All right, I'll talk about viruses next in the other video.